This is Chapter 4 of the Instructor's video manual for cloning a fluorescent gene. In today's lab, your students will be tricking E. coli bacteria to take in their recombinant DNA plasmid from a solution, incorporating it and using its genetic code to make proteins. Today's lab is the longest of all of the 6 CAFG labs, so you're going to have to do a little bit of work to be prepared before your students come into the lab. You're going to need a 42 degrees Celsius water bath, which is going to be used for transformation of the E. coli bacterial cells. You're going to need ice buckets that the students can put their bacteria on to cool them down before heat shocking. Today will also require a few new reagents. The frozen E. coli bacteria, which should have been in the freezer in the original packaging that we've sent in our kit until now, can be taken out and thawed in an ice bucket. It's really, really important to keep these bacteria cold, because if they become unfrozen even once, they reduce their competence about 3-fold. And every day that they're left unfrozen in a refrigerator, they lose about half of their competency again. We're also going to need SOX buffer, SOCS. This is a very, very rich bacterial growth medium, that the cells are going to use to help recover themselves from the stress of being heat shocked during a little bit of time that we're going to give the cells to start producing the antibiotic resistance protein before we plate the cells onto the ampicillin plates that have an antibiotic on them. Finally, we're going to need to bring out transfer pipettes. These are the little bulb pipettes that are demonstrated in the video to go along with Chapter 4. All 3 of these tools should be placed at an accessible location, because all of the students are going to be running up to the instructor's bench, or wherever all of these things are, and grabbing their transfer pipettes, and grabbing their SOX broth, and heat shocking their cells, and there's going to be a lot of traffic coming into the central instructor's bench location, because they haven't aliquoted out all of their reagents like they did for the first couple labs. We also recommend that you leave at least one day free between Chapter 4 and Chapter 5 of CAFG. This is because Chapter 4 works best if you can leave the transformed cells in the incubator recovering overnight, or at least for 2 hours, and then have the students come back and plate the cells onto their ampicillin and arabinose plates at the end of the day or at the beginning of the next school day. After plating, the bacterial cells take at least 18, sometimes as much as 24 hours to grow up and form colonies. That means you don't really want to cut it too close and end up in a situation where you've planned to do Chapter 5, but the colonies probably need another day to grow to become large enough to see and experience, and then having to, rush and get a new lesson plan put together and swap places with Chapter 5. Even though we say that Chapter 4 and Chapter 5 should be spaced at least one day apart, you don't want to let the plates sit in the incubator with the colonies growing in them for too long. That's because the beta-lactamase from the antibiotic-resistant colonies is actually secreted out into the agar, where it chews up and neutralizes all of the ampicillin in the plates. This means that bacteria not containing any plasmid can grow up around a successfully transformed plasmid, because all of the ampicillin in that area has been eaten up by the transformed colony. Once you see that the colonies on the plates have started growing up, and you can visually see them, just take the plates and move them into the refrigerator. That'll stop the colonies from growing any further and inhibit the growth of any of these satellite colonies that don't contain plasmid. The plates will be just fine in the refrigerator for at least a week, but if you leave them longer than that, some of them will start to develop mold, since most molds are ampicillin resistant.